Hi everyone. Today is the 10th problem solving session for the course Overview and Integration of Cellular Metabolism. The course instructors are Professor Aritri Veer and Professor Arindam Ghosh. And I'm Shrija Ghosh, a PMRF from IIT Kanpur. I'm here to help you with the solving of the assignment questions. So today we'll discuss the questions related to heme synthesis, basically heme metabolism, both synthesis and its catabolism, disorders related to heme metabolism. Then we'll discuss bilirubin metabolism and disorders related to that. And we'll start with the nucleotide synthesis, where we'll start with the purine synthesis. Okay, so I have prepared certain questions related to all these topics and hope to cover all the topics discussed in the in week 10 session through different questions. These questions will help you solve your own assignment questions, and if you have any doubt, feel free to ask me. Okay. So starting with the first question, what is the first step in heme synthesis? Option A, condensation of glycine and succinyl coenzyme A to form delta ALA. Option B, decarboxylation of uroporphyrinogen 3. Option C, insertion of iron into protoporphyrin. Option D, formation of porphobilinogen. What is the first step in the heme synthesis? Option A, condensation of glycine and succinyl coenzyme A to form delta ALA. Option B, decarboxylation of uroporphyrinogen 3. Option C, insertion of iron into protoporphyrin. And option D, formation of porphobilinogen. So what do you think would be the correct answer? So the correct answer is option A. Just a second. Yeah. So the correct answer is option A, condensation of glycine and succinyl coenzyme A to delta ALA. So this is the first step in the heme synthesis process where ALA is synthesized. ALA is delta amino levulinic acid. So in most animals, including mammals, the synthesis starts with the condensation of succinyl coenzyme A and glycine in the presence of pyridoxal phosphate to form delta amino levulinic acid, that is ALA, delta ALA. Okay. So this is the first step in the heme synthesis process where succinyl coenzyme A and glycine, they both condense together, they both combine together, and the coenzyme A gets released, and the intermediate that is formed is alpha amino beta keto adipate. This alpha amino beta keto adipate, uh, after decarboxylation, becomes delta amino levulinate or delta amino levulinic acid. Okay. This reaction is catalyzed by the enzyme delta amino levulinate synthase. Since delta amino levulinate is being synthesized, the enzyme's name is delta amino levulinate synthase. And noted down that it is synthase and not synthetase. I've told you in the previous session also that if it is a synthetase, that means ATP hydrolysis is required. If it is a synthase, then there's no ATP hydrolysis required in this. So since this is a synthase, we don't require any ATP hydrolysis, and uh, this reaction can proceed just by the condensation of succinyl coenzyme A and glycine. So the intermediate will form after the coenzyme A removal, that is alpha amino beta keto adipate, and then decarboxylation of this intermediate. Decarboxylation of this intermediate will result in delta amino levulinic acid. Okay. So in case of pyridoxine deficiency, anemia may be manifested because this reaction uh, occurs in the presence of pyridoxal phosphate. Okay, so this enzyme ALA synthase, it works along with pyridoxal phosphate. So if there is a pyridoxine deficiency, there could be anemia because you know that heme synthesis is important for the production of RBCs. So anemia may be manifested in case heme synthesis is defective. So this is the first step of heme synthesis. If this is defective, obviously there's no heme that is going to be synthesized. And this enzyme ALA synthase, it is located in the mitochondria and it is rate limiting enzyme of the pathway. Okay. I hope that's clear to everybody. Moving on to the next question. What is the precursor of delta amino levulinic acid in bacteria and plants? What is the precursor of delta amino levulinic acid in bacteria and plants? Option A, succinyl coenzyme A. Option B, glutamate. Option C, arginine. Option D, glycine. What is the precursor of delta amino levulinic acid in bacteria and plants? Succinyl coenzyme A, glutamate, arginine, and glycine. What is the precursor of delta amino levulinic acid in bacteria and plants? Option A, succinyl coenzyme A. Option B, glutamate. Option C, arginine. And option D, glycine. What do you think would be the correct answer? So the correct answer is option B, glutamate. So we saw in most animals, the delta amino levulinic acid or delta ALA is synthesized from the condensation of succinyl coenzyme A and glycine, right? But in bacteria and plants, the delta amino levulinic acid is synthesized from glutamate. Okay, so we'll see how. So we saw this path uh, in the previous question. We saw succinyl coenzyme and glycine condensation, right? But in bacteria and plants, from glutamate, delta amino levulinic acid or delta amino levulinate is synthesized. How? From glutamate, one tRNA uh, gets attached to the glutamate to become glutamyl tRNA. This glutamyl tRNA, uh, with the help of glutamyl tRNA synthetase, now again, see, this is a synthetase. That means ATP hydrolysis is required. And this is a high energy requiring reaction. So this ATP is hydrolyzed into AMP and pyrophosphate is released. And this drives the energy of this drives the reaction forward. Basically, this provides the energy for the reaction to go ahead. So tRNA, which is specific for glutamate, gets attached to the glutamate, forming glutamyl tRNA. And this glutamyl tRNA by the enzyme glutamyl tRNA reductase will be reduced to glutamate 1 semialdehyde. And this enzyme requires one molecule of NADPH. NADPH is oxidized to NADP plus, and glutamyl tRNA is reduced to glutamate 1 semialdehyde, and the tRNA is released. This tRNA could be recycled, and this glutamate 1 semialdehyde is then converted to delta amino levulinate by the enzyme glutamate 1 semialdehyde aminomutase. So you see this uh, so this molecule and this molecule is basically isomers. So if you see the molecular formula for both the molecules is same, it's just that the amino group is transferred from this carbon to the last carbon, and the uh, carbon that was having the aldehyde group, this aldehyde group now shifted to the second last carbon, making it a uh, ketone group. So it is having a amino and acid group along with a keto group. Okay. So glutamate, one semialdehyde, amino mutase. Since the amino group is moving from one carbon to the another, it is the enzyme is named as amino mutase. Okay. Mutases are the enzymes that can actually convert one isomer to into another. Okay. So from glutamate, in bacteria and plants, from glutamate, this delta ALA is synthesized first by glutamyl tRNA synthetase, where glutamate is attached to the tRNA, and in that process, one ATP is hydrolyzed into AMP and a pyrophosphate is released. And then glutamyl tRNA is reduced to glutamate 1 semialdehyde, and then glutamate 1 semialdehyde is converted to delta ALA by glutamate 1 semialdehyde amino mutase. Glutamyl tRNA to glutamate 1 semialdehyde is carried out by the enzyme glutamyl tRNA reductase, where one NADPH molecule is oxidized to NADP plus. Okay, I hope this is clear. Moving to the next question now. Porphobilinogen is formed by the condensation of option A, two ALA molecules, option B, two glycine molecules, option C, two succinyl coenzyme A molecule, option D, glycine and proline. Porphobilinogen is formed by the condensation of option A, two ALA molecules, that is alpha levulinic acid, amino levulinic acid. Option B, two glycine molecules. Option C, two succinyl coenzyme A molecules. And option D, glycine and proline. Porphobilinogen is formed by the condensation of. What would be the correct answer? The correct answer is option A, two ALA molecules, so two amino levulinic acid molecules. So two ALA condenses to form porphobilinogen. This is basically the second step in the heme synthesis. So we saw the synthesis of amino levulinate in the first step, right? In the first step of heme synthesis, we got delta amino levulinate. Now from this delta amino levulinate, two molecules of delta amino levulinate can condense to form one molecule of porphobilinogen. So in this reaction, you can see it is given 8 molecules of delta amino levulinate. So basically, if two molecules of ALA is giving one molecule of porphobilinogen, that means 8 molecules will give 4 molecules of porphobilinogen, right? So this reaction is carried out by porphobilinogen synthase, and two molecules of water is removed during the condensation process. And this reaction occurs in the cytoplasm. Okay. So two molecules of ALA are condensed to form porphobilinogen, that is abbreviated as PBG. And in this process, two molecules of water are removed. So here you can see for 8 molecules of ALA, 8 molecules of water are removed. Okay. And the enzyme that catalyzes this reaction is called PBG synthase, porphobilinogen synthase, or ALA dehydratase, because dehydration reaction is happening, right? H2O is released. So it's a dehydration reaction. the water molecules are removed, so that is a dehydration reaction, and the enzyme that would be required for this reaction would be a dehydratase. So ALA dehydratase, since the H2O is removed from ALA, it is ALA dehydratase, or it is also known as PBG synthase, since synthesis of PBG is occurring. Okay, PBG is porphobilinogen, and this is a monopyrrole. So you see this ring structure is called pyrrole ring. Okay, this ring structure, it is called pyrrole ring. And since there's only one ring structure, it is called monopyrrole. Porphobilinogen is a monopyrrole. The enzyme contains zinc and is inhibited by lead. This enzyme, it contains zinc and it is inhibited by lead. Okay. So this is the second step in the heme synthesis. From delta aminolevulinate, two molecules of delta aminolevulinate, we get one molecule of porphobilinogen, where the cyclization is occurring by the enzyme PBG synthase or ALA dehydratase. And for two molecules of ALA, two molecules of water is removed. And this PBG is a monopyrrole since it has one pyrrole ring structure. Okay. I hope that's clear. Moving on to the next question. Which enzyme is required for the conversion of protoporphyrin to heme? Which enzyme is required for the conversion of protoporphyrin to heme? Option A, protoporphyrinogen oxidase. Option B, delta aminolevulinate synthase. Option C, delta aminolevulinate dehydratase. And option D, ferrochelatase. Which enzyme is required for the conversion of protoporphyrin to heme? Option A is protoporphyrinogen oxidase. Option B is delta aminolevulinate synthase. Option C is delta aminolevulinate dehydratase. And option D is ferrochelatase. Which enzyme is required for the conversion of protoporphyrin to heme? Protoporphyrinogen oxidase, delta aminolevulinate synthase, delta aminolevulinate dehydratase, or ferrochelatase. What do you think would be the correct answer? The correct answer is option D, ferrochelatase. So this is the last step in the heme formation or heme synthesis, where the ferrous ion is introduced or attached to the protoporphyrin. So the whole porphyrin ring is formed with four pyrrole ring structure. So I told you this is a pyrrole ring structure, and now four pyrrole ring structures have are connected to each other, and this whole structure is called protoporphyrin. Protoporphyrin is basically one step before the heme synthesis occurs. Now in this, iron molecule needs to be introduced for the conjugation with these nitrogen, and that iron containing protoporphyrin is called heme. Okay, so the enzyme required for this synthesis is called heme synthase, since heme is being synthesized, or it is also known as ferrochelatase, and this enzyme is located in mitochondria. Remember, the location of each enzyme is very important. So if you remember from the first step onwards, first step where ALA was being synthesized, ALA synthase, that was present in mitochondria. The second step, ALA dehydratase, that enzyme was present in cytosol. Okay, and this is the last step where again ferrochelatase, this enzyme is also present in mitochondria. So you have to remember which enzyme is present in mitochondria and which enzyme is present in cytosol, because that is how you will remember which reaction is taking place inside the mitochondria and which reaction is taking place outside the mitochondria in the cytosol. Okay. So heme synthesis is ultimately occurring in the mitochondria. That means, okay, final heme product is formed inside the mitochondria because this enzyme heme synthase or ferrochelatase, this is present in the mitochondria. Okay, so Fe2 plus is introduced or added or attached to the protoporphyrin to form heme. So this is the whole synthesis of heme. So I told you the first step begins with succinyl coenzyme and glycine. They both condense together with the help of delta aminolevulinate synthase, delta ALA synthase, to give the product delta aminolevulinate. Okay, delta ALA we get. After this, two molecules of delta ALA will condense together to form porphobilinogen by the enzyme ALA dehydratase. Okay, two molecules of ALA will combine to give porphobilinogen. Now, four molecules of porphobilinogen together will form uroporphyrinogen. Okay. So you remember porphobilinogen had one pyrrole ring, right? So, and the final heme has four pyrrole rings. So, four molecules of porphobilinogen together forms uroporphyrinogen 3 by the enzyme uroporphyrinogen 1 synthase or uroporphyrinogen 3 co-synthase. Then decarboxylation occurs with the enzyme uroporphyrinogen decarboxylase, where the product is coproporphyrinogen 3. And this coproporphyrinogen 3, it gets oxidized by coproporphyrinogen oxidase, where CO2 is removed to give protoporphyrinogen 3. And from protoporphyrinogen 3, again one oxidation step occurs by the enzyme protoporphyrinogen oxidase to give protoporphyrin 9. And this protoporphyrin 9 by the enzyme ferrochelatase or heme synthase, the Fe2 plus is attached and we get the final product that is heme. Okay. So you see the reaction begins with succinyl coenzyme and glycine. The condensation of both results in delta ALA, which is which will then form the pyrrole ring structure in the porphobilinogen. Then one porphobilinogen has one pyrrole ring structure. So four such molecules will come together to make the porphyrin ring structure, the basic structure of porphyrin. So that is called the uroporphyrinogen 3. Then we'll get the coproporphyrinogen 3 and then protoporphyrinogen 3. And finally, we'll get the protoporphyrin 9, which is the heme without the iron, ferrous iron. So the ferrous iron will be attached to protoporphyrin 9 to form heme by the enzyme ferrochelatase. Okay. I hope this is clear. Moving to the next question then. Excess heme regulates its own synthesis by option A, stimulating ALA synthase activity. Option B, inhibiting ALA synthase gene expression. Option C, activating ferrochelatase. And option D, none of the above. Excess heme regulates its own synthesis by option A, stimulating the ALA synthase activity. Option B, inhibiting ALA synthase gene expression. Option C, activating ferrochelatase. And option D, none of the above. What is the correct answer? The correct answer is option B, inhibiting ALA synthase gene expression. So this is what I was talking about when I said that you should remember which enzyme is present in mitochondria and which enzymes are present in cytosol, because this compartmentalization actually helps in the regulation of heme synthesis. And when excess heme is produced, it directly inhibits the ALA synthase, which is the first uh, enzyme in the pathway. So if it inhibits ALA synthase, the cycle will stop there. If the body has enough heme and it wants to signal now that we don't need more heme synthesis, it will directly stop it at the first step. It will inhibit the ALA synthase gene expression. Okay. So this is possible because of this compartmentalization. So you see here ALA synthase is present in mitochondria, where ALA is formed. Then ALA is transported into the cytosol, and the porphobilinogen, uroporphyrinogen 3, and coproporphyrinogen 3 synthesis occurs in the cytosol. So all the enzymes associated with these steps are present in the cytosol. After this, again the reactions occur in the mitochondria. So protoporphyrinogen 3 formation and the protoporphyrin 3 formation occurs in the mitochondria, and the ferrochelatase, if you remember, I told you that enzyme is also present in mitochondria. So the iron or the ferrous ion insertion in the protoporphyrin occurs in mitochondria. So the heme is forming in the mitochondria, and that ALA synthase is also in the mitochondria. So it is easier for the heme molecule, if excess heme is produced in the mitochondria, it is easier for those heme molecules to go and inhibit the ALA synthase gene expression, because that is also present in the mitochondria. Okay, and this is a type of feedback inhibition where the product is inhibiting its synthesizing enzymes, so that when excess of product is forming, the cycle can be seized. Okay. So this is how part of the heme synthesis occurs in mitochondria and part of the heme synthesis occurs in cytosol. And this compartmentalization actually helps in regulation of heme synthesis. When excess of heme is synthesized, it inhibits the ALA synthase gene expression or ALA synthase activity, which is the first step in the heme synthesis. Okay. Next question is, which enzyme is inhibited by lead poisoning in heme synthesis? Which enzyme is inhibited by lead poisoning in heme synthesis? Option A, ALA synthase. Option B, ferrochelatase. Option C, ALA dehydratase. And option D, both B and C. Which enzyme is inhibited by lead poisoning in heme synthesis? Option A, ALA synthase. Option B, ferrochelatase. Option C, ALA dehydratase. And option D, both B and C. Which enzyme is inhibited by lead poisoning in the heme synthesis? ALA synthase, ferrochelatase, ALA dehydratase, and both B and C. What do you think would be the correct answer? The correct answer is option D, both B and C. That is, both ferrochelatase and ALA dehydratase are inhibited by lead poisoning during heme synthesis. So, during heme synthesis, if there's lead poisoning, it can inhibit both ferrochelatase, which is responsible for the ferrous ion attachment to the protoporphyrin, and ALA dehydratase, which is responsible for the uh, formation, which is responsible for the formation of porphobilinogen from ALA. Okay. The next question is, which is not a heme protein? Which of the following is not a heme protein? Option A, myoglobin. Option B, cytochrome P450. Option C, hemoglobin. And option D, immunoglobulin G. Which of the following is not a heme protein? Okay. You have to tell which protein is not containing the heme. Option A, myoglobin. Option B, cytochrome P450. Option C, hemoglobin. And option D, immunoglobulin G. So the correct answer is option D, immunoglobulin G. So rest all the three proteins, myoglobin, cytochrome P450s and hemoglobin, all of them contain the heme in their active center. So these are some of the proteins that contain heme. So hemoglobin is the first one. It contains heme. It is present in the red blood cells that gives the red color to our blood. So heme is red in color. Okay. So all these proteins will give a reddish color due to the presence of heme. Okay. So hemoglobin, it is present in the red blood cells. It is our blood is red in color, you know. Myoglobin also contains the heme. Then cytochrome P450s, they contain heme in their active center, and that catalyzes the oxidation reactions. Peroxidases are also present, which contain heme as an active center, and again they perform the oxidation reaction. Then there are catalase which can contain heme in their active center. Tryptophan pyrrolase. There's a protein enzyme called tryptophan pyrrolase that contains heme in their active center. And then nitric oxide synthase. It also contains heme in their active center. Okay, so these are the list of proteins which can contain heme in their active center. And all these proteins will have a reddish tinge because of the presence of heme in their active center. Okay. Moving on to the next question now. Porphyrias are due to option A, defective heme catabolism. Option B, defect in heme synthesis. Option C, increased bilirubin conjugation. And option D, excess dietary iron. Porphyrias are due to option A, defective heme catabolism. Option B, defect in heme synthesis. Option C, increased bilirubin conjugation. And option D, excess dietary iron. So the correct answer is option B, defect in heme synthesis. Porphyrias, it's a disorder, and these disorders are associated with the biosynthesis of heme. So if there's a defect in the heme synthesis, heme biosynthesis, so we saw the heme synthesis pathway, right, where it was starting from succinyl coenzyme and glycine, and finally we were getting the product heme. So in that biosynthetic pathway, if there's a disorder, if there's a defect, for the disorders that can arise due to that different defects in the biosynthesis of heme in that pathway, such disorders are called porphyrias. And this can arise due to mutations of genes coding for the enzymes in the heme synthesis. So you saw multiple enzymes are there in the heme synthesis pathway, right? So there could be mutations in any of those genes coding for the enzymes. So any of those enzymes could be defective, leading to different types of porphyrias. And how do we Identify porphyria. Porphyria, it is characterized by increased production and excretion of porphyrins or their precursors. So, if heme synthesis is defective, that means either we'll get ALA excretion, because after ALA, the porphyrin ring starts to form, right? The pyrrole formation will occur and then the porphyrin formation will occur. So if there's a defect in the downstream genes of the heme synthesis pathway, then there might be a chance of excretion of either porphyrins, all those uh, protoporphyrinogen and coproporphyrinogen, all those porphyrin rings, which are the precursors of heme, those could be excreted, or ALA and PBG, which are before the porphyrin ring formation occurs, the precursors which are there, those could be excreted. So anything that is there in the pathway before heme could be excreted depending on which enzyme is defective. So if the uh, enzyme that is converting PBG into the porphyrinogen that is defective, then PBG will be excreted in the urine. Then if the defect is in ferrochelatase, then protoporphyrin will be excreted. So depending on which enzyme is defective, different porphyrins or their precursors could be excreted, and that is the characteristic of porphyrias. Most of the porphyrias are inherited as autosomal dominant traits. So these are genetic disorders. And I told you this is due to the mutation of the genes encoding the enzymes in the heme synthesis pathway. So, and this genetic disorder is due to, or most of them are autosomal dominant traits. Okay. Moving to the next question. Which porphyria is associated with the deficiency of the enzyme porphobilinogen deaminase? Porphobilinogen deaminase. The deficiency of this enzyme is associated with which porphyria? Option A, acute intermittent porphyria. Option B, porphyria cutanea tarda. Option C, congenital erythropoietic porphyria. Option D, hereditary coproporphyria. And option E, X-linked dominant protoporphyria. Which porphyria is associated with the deficiency of the enzyme porphobilinogen deaminase? Which porphyria is associated with the deficiency of the enzyme porphobilinogen deaminase? Option A, acute intermittent porphyria. Option B, porphyria cutanea tarda. Option C, congenital erythropoietic porphyria. Option D, hereditary coproporphyria. And option E, X-linked dominant protoporphyria. So the correct answer is option A, acute intermittent porphyria. So acute intermittent porphyria is caused due to the porphobilinogen deaminase deficiency. So you remember porphobilinogen deaminase, it was converting porphobilinogen into the uh, protoporphyrin porphyrinogen ring, right? So four molecules of porphobilinogen were condensing together, because this is a monopyrrole and four pyrrole ring structure. The porphyrin ring structure starts forming. Okay. So the enzyme PBG deaminase, the deficiency of this enzyme, which leads to the deficiency of this enzyme causes acute intermittent porphyria. So porphobilinogen will not be able to get converted into uroporphyrinogen. And there's a defect in the gene that synthesizes this enzyme that leads to acute intermittent porphyria. And as a result, the secondary increase in the activity of ALA synthase occurs because there's no end product inhibition. So if you remember from the regulation of heme synthesis, the excess heme by feedback inhibition, the excess heme inhibits the ALA synthase, right? Now, if there's a defect in PBG deaminase and the pathway cannot go further, that means there's no heme production, and that would lead to increase in the ALA synthase activity because there's nothing to inhibit the ALA synthase, right? Do you understand? So there's no molecule that can inhibit ALA synthase because heme was going to inhibit the ALA synthase, and now that heme synthesis is not happening, increased activity of ALA synthase occurs, and levels of both ALA, since ALA synthase activity is increased, so levels of ALA and PBG both are elevated in the blood and urine. Okay, so this is acute intermittent porphyria, which is caused by porphobilinogen deaminase deficiency or defect. And this defect or deficiency is due to the defect in the gene that codes for this enzyme. This enzyme is also known as uroporphyrinogen 1 synthase or hydroxymethylbilane synthase. Okay. I hope that's clear. And in acute intermittent porphyria, levels of ALA and PBG are elevated in the blood and urine because ALA synthase activity is highly increased because of the absence of heme that can inhibit the ALA synthase by feedback inhibition. Okay. The next question is, which enzyme is defective in the erythropoietic protoporphyria? Which enzyme is defective in the erythropoietic protoporphyria? Option A, protoporphyrinogen oxidase. Option B, ferrochelatase. Option C, ALA synthase. And option D, PBG deaminase. Which enzyme is defective in erythropoietic or protoporphyria? Option A, protoporphyrinogen oxidase. Option B, ferrochelatase. Option C, ALA synthase. And option D, PBG deaminase. Which enzyme is defective in erythropoietic protoporphyria? Option A, protoporphyrinogen oxidase. Option B, ferrochelatase. Option C, ALA synthase. Option D, PBG deaminase. The correct answer is option B, ferrochelatase. So you saw in the previous question, PBG deaminase was defective and it was leading to acute intermittent porphyria, AIP. In this case, defect in another enzyme in the heme synthesis, that is ferrochelatase, which is the last enzyme. It leads to erythropoietic protoporphyria. So ferrochelatase enzyme is partially active in case of erythropoietic protoporphyria. Now, this enzyme is responsible for the iron attachment, ferrous iron in attachment to the protoporphyrin. So if you remember the role of ferrochelatase, it was in doing the iron insertion in the porphyrin ring, right? Now, since there won't be iron insertion, ferrochelatase is defective, so there won't be any heme synthesis. And as a result, the substrate, that is protoporphyrin 9, it will be in excess because it is not getting converted to heme. So this excess protoporphyrin in plasma and erythrocytes is present. So basically the levels of protoporphyrin in plasma and erythrocyte will increase, and it will be excreted uh, in the feces also. Okay, so naturally the substrate, that is substrate of ferrochelatase, that is protoporphyrin, it will keep getting accumulated and it will be eventually excreted. Okay, and excretion of porphyrin or its precursors in urine is also normal in such cases. And the clinical symptoms of photosensitivity, it is a major symptom, okay? Photosensitivity. In case of erythropoietic protoporphyria, photosensitivity is a major symptom. Apart from that, liver cirrhosis and anemia can occur. Anemia because, again, heme synthesis is not occurring. So any defect in heme synthesis could lead to anemia. Okay, I hope that's clear. Moving to the next question. Porphyrias can be caused by option A, genetic mutations, option B, lead poisoning, option C, both, or option D, neither. Porphyrias can be caused by option A, genetic mutations, option B, lead poisoning, option C, both, and option D, neither. So the correct answer is option C, both. So porphyrias can be caused by both genetic mutation and lead poisoning. Okay. So both genetic mutation and lead poisoning can cause porphyria. So defect in the heme synthesis could be due to genetic reasons, that there is some defect in the genes that are coding for the enzymes present in the heme synthesis, or lead poisoning. So if you remember, lead poisoning was in we saw in the one of the previous questions that lead poisoning could cause inhibition of ferrochelatase as well as ALA dehydratase, right? So lead poisoning can also cause defective heme synthesis, uh, and genetic and obviously genetic reasons could be the reason behind defective heme synthesis. So both the reasons could cause porphyria in human. Okay. The next question is, lead poisoning can cause option A, acute porphyria, option B, acquired porphyria, option C, jaundice, and option D, none. Lead poisoning can cause option A, acute porphyria, option B, acquired porphyria, option C, jaundice, and option D, none. So the correct answer is option B, acquired porphyria. So acquired porphyria, by the name itself, you can see it is acquired. So you have different types of porphyria. You have the porphyria that is due to genetic reasons, and then you have acquired porphyria that is due to lead poisoning. Okay, and I've told you lead poisoning can lead to inhibition of ferrochelatase and ALA dehydratase. Now, both of these enzymes, ferrochelatase and ALA dehydratase, they are present in heme synthesis pathway, right? So ferrochelatase is the enzyme that is present in the last step of heme synthesis, and ALA dehydratase is the enzyme that is present at the second stage of heme synthesis. Inhibition of both of these enzymes can lead to defective heme synthesis. Lead can actually displace the zinc ion at the enzyme's active site. So if you remember, ALA dehydratase has the zinc ions in the active site, right? So lead can replace that zinc ion, leading to inhibition of the enzyme. Excretion of porphyrins in the urine is up to 10 mg per day. So porphyria, I told you, is characterized by the excretion of porphyrin. And in case of acquired porphyria, which is due to the lead poisoning, the excretion could be uh, up to 10 mg per day. Excretion of porphyrins in urine could be 10 mg per day. And automobile exhaust is a common source for lead. So basically the pollution that is caused by automobiles, that is the common source for lead. So if you are inhaling the exhaust coming out of the automobiles, you could get the lead poisoning, which could lead to porphyria. In children, it can cause developmental defects, drop in IQ, hyperactivity, insomnia, and many other health problems. And in adults, it can cause severe abdominal pain, mental confusion, and many other symptoms related to mental health. And drugs, there are certain drugs that can cause porphyria. Uh, that include steroids, oral contraceptives, barbiturates, and pesticides. Okay, so anything that can contain uh, low levels of lead, and if it enters your body, lead can actually inhibit the enzymes in the heme synthesis biosynthesis pathway, and that can lead to defective heme synthesis and excretion of porphyrins in the urine, leading to acquired porphyria. I hope that's clear. The next question is, which porphyria is not considered acute? Which porphyria is not considered acute? Option A, hereditary coproporphyria. Option B, variegate porphyria. Option C, porphyria cutanea tarda. And option D, acute intermittent porphyria. Which porphyria is not considered acute? Hereditary coproporphyria, variegate porphyria, porphyria cutanea tarda, and acute intermittent porphyria. The correct answer is option C, porphyria cutanea tarda, that is PCT. So there are two types of porphyrias, acute porphyria and chronic porphyrias. So acute porphyria includes acute intermittent porphyria, AIP, variegate porphyria, VP, aminolevulinic acid dehydratase deficiency porphyria, that is ALAD, and hereditary coproporphyria, that is HCP. Okay, so acute porphyria, it includes all these four types of porphyria. These diseases primarily affect the nervous system, resulting in episodic crises known as acute attacks. The major symptoms of an acute attack is an abdominal pain, often accompanied by vomiting, hypertension, elevated blood pressure, and tachycardia and abnormally rapid heart rate. So, acute porphyria, it can affect the nervous system, and it can have multiple major symptoms of acute attack is abdominal pain, and it is accompanied by vomiting, hypertension, and sometimes rapid heart rate. Okay, rapid increase in the heart rate. Then comes the second type of porphyria, that is chronic porphyria. Okay, the non-acute porphyria, the chronic porphyria. It includes X-linked dominant protoporphyria, XLDPP, X-linked dominant protoporphyria. So XLDPP. Second is congenital erythropoietic porphyria, CEP. Third is porphyria cutanea tarda, that is PCT. And fourth is erythropoietic protoporphyria, EPP. Okay, so there are four types of chronic porphyrias and there are four types of acute porphyrias. Okay, so you should remember which one is what. And none of these chronic porphyrias are associated with acute attacks. Their primary manifestation is with the skin disease. For this reason, these four porphyrias, along with two acute porphyria, that is the variegate porphyria VP and hereditary coproporphyria HCP,
the because VP and HCP can also include the skin manifestation. They are sometimes called cutaneous porphyria. Okay, all these forms of porphyria, all the chronic porphyrias, along with two of the acute porphyrias, VP and HCP, they are collectively called cutaneous porphyria because they mainly cause the skin disease as a manifestation. Okay, manifestation of these chronic porphyria is mainly skin disease. There's no acute attacks and all that are present during acute porphyrias. Okay, I hope that's clear to everybody. Moving to the next question now. Heme is degraded initially by. Heme is degraded initially by. So till now we were talking about heme synthesis. Now we come to heme degradation. Heme is degraded initially by option A, heme oxygenase, option B, biliverdin reductase, option C, glucuronil transferase, and option D, ferrochelatase. Heme is degraded initially by heme oxygenase, biliverdin reductase, glucuronil transferase, and ferrochelatase. The correct answer is heme oxygenase. So heme is degraded by the enzyme heme oxygenase. So you know heme is a porphyrin. Heme has a porphyrin ring structure with the iron inside it, and when the heme starts degrading by the enzyme heme oxygenase, first step is the ring opening. So at this point where alpha is written, at this point the ring opens. And then if you see these methyl groups, vinyl groups, and propionyl groups are marked, right? So you can see when the ring opens from here, you have a double bond O here, and then this pyrrole ring with the methyl and vinyl group. And then after the vinyl group, we have this double bond and then single bond here, double bond and single bond. And then again the methyl group and propionyl group. And then again the pyrrole ring is there. And if you see, the whole structure is there. So just the ring opens from here, the iron comes out of it, and one carbon monoxide comes out of the heme structure. And the product that we get, this open ring pyrrole structure, this is called biliverdin. Okay, so heme gets converted to biliverdin by the enzyme heme oxygenase. So heme gets converted to biliverdin by the enzyme heme oxygenase. The yellow pigment of serum after hemolysis is due to option A bilirubin, option B biliverdin, option C urobilinogen, and option D myoglobin. The yellow pigment of serum after hemolysis is due to option A bilirubin, option B biliverdin, option C urobilinogen, and option D myoglobin. The correct answer is option A, bilirubin. So bilirubin is present in the serum, and it gives the yellow color to the serum. So if you have seen the serum separated from blood in any of the class experiments, you would have seen that yellowish color, and that yellowish color is due to the presence of bilirubin. Okay. Moving on to the next question. The unconjugated bilirubin in blood is mostly carried by option A, it is carried in the free form. Option B, it is bound to albumin. Option C, it is bound to hemoglobin. And option D, it is carried as conjugated bilirubin. The unconjugated bilirubin in blood is mostly carried by free form, bound to albumin, bound to hemoglobin, or as a conjugated bilirubin. What would be the correct answer? The correct answer is option B, bound to albumin. So bilirubin in itself is insoluble in water. Okay. So heme is degraded into biliverdin. We saw the reaction by heme oxygenase. The heme ring structure is opened, and we get the compound called biliverdin. That biliverdin is converted to bilirubin. All this is occurring in the reticuloendothelial system. Okay. So from the RBCs, when RBC lies, the heme comes out. The heme will get converted to biliverdin and then bilirubin. This bilirubin is insoluble in water. Okay. So for it to be transported across through our system, it needs to be soluble, right? Insoluble matter cannot uh, get transported through blood or uh, so for bilirubin to be transported to liver, it needs to be conjugated to another protein called albumin. So albumin is present in the plasma, and when bilirubin is released in the plasma, it conjugates with the albumin to form a bilirubin albumin complex. So all this is occurring for the disposal of bilirubin. Okay, so bilirubin has no function in our body, but it is a product that is formed due to the degradation of heme, and it needs to be excreted out of the body. But for something to be excreted out of the body, it needs to be solubilized first. Okay, so liver plays the central role in the disposal of bilirubin. Bilirubin formed is insoluble in water, I told you, and this lipophilic bilirubin is therefore transported in plasma bound to albumin. So, albumin is another protein through to which this bilirubin is bound, uh, and its albumin is a serum protein. Okay, it's called serum albumin. Okay, you might have heard about bovine serum albumin (BSA) for protein estimation if you have read about it. That so humans also have albumin in their serum. It is human serum albumin, and in uh, with that albumin, this bilirubin binds and becomes soluble. Basically, albumin helps the bilirubin to albumin basically carries the bilirubin in the plasma in the serum so that it could be transported to the liver. Okay. Now, one molecule of albumin can bind two molecules of bilirubin, and 100 ml of plasma can transport up to 25 mg of bilirubin. Okay. So, it's a great excretion process. Albumin binds the bilirubin in loose combination because it has to release the bilirubin later. Okay. So, it can't be tightly bound to the albumin. And because of this loose combination, when present in excess, bilirubin can actually dissociate from the albumin also. And binding site for bilirubin on albumin can be occupied by aspirin, penicillin. So penicillin is an antibiotic. Aspirin is a very common drug that we take during headache. And we should be careful while taking all these drugs and antibiotics because See, aspirin, penicillin, they can bind to albumin at the same binding site where bilirubin is binding. So if we have excess of aspirin and penicillin in our body, it can actually dissociate the bilirubin. And binding of bilirubin to albumin is very important for its excretion. Otherwise, bilirubin accumulation will start occurring in our body, which is not good for the body. Okay. So such drugs can therefore displace bilirubin from albumin. Hence, care should be taken while administering such drugs to newborn babies to avoid kernicterus. Now, kernicterus is a condition where bilirubin accumulation starts occurring, and when excess bilirubin is present and it has nowhere to go, it somehow finds a way to cross the blood brain barrier, and which is a very serious condition, because if bilirubin crosses the blood brain barrier, that means it can enter the brain now, and it can cause symptoms like encephalopathy and all. Okay. So that's why the transport of bilirubin is very important, and for that, conjugation of bilirubin to albumin is very important for it to be solubilized or carried into the plasma. Moving to the next question, bilirubin is finally excreted as. Bilirubin is finally excreted as option A, biliverdin, option B, urobilin, option C, stercobilin, and option D, porphyrins. Bilirubin is finally excreted as. So multiple answers could be correct. Option A, biliverdin, option B, urobilin, option C, stercobilin, and option D, porphyrins. So the correct answer is option B and option C, urobilin and stercobilin. So in this excretion process, the final excretion process, the bilirubin, which was bound to which was bound to albumin, it is carried to the liver, and from the liver, uh, it gets converted into bilirubin diglucuronide, and it enters the bile ducts. From the bile ducts, small intestine and large intestine, it gets converted into urobilinogen and stercobilinogen. So urobilinogen gets transported to kidney, and stercobilinogen is present in the intestine. Stercobilinogen gets converted into stercobilin, and urobilinogen in the kidney gets converted into urobilin. So stercobilin gets excreted in the feces, and urobilin gets excreted in the urine. Okay, so UBG, that is urobilinogen, and SBG, that is stercobilinogen, are both colorless compounds, but they are oxidized to colored compounds, urobilin and stercobilin, respectively. So urobilin has a yellowish color that gives the yellow color to your urine, and stercobilin has a brownish color that gives the color to the feces. Okay. So this is the final excretion process. This is how bilirubin is excreted out of our body. So when RBCs are lysed and the heme is out and open, now heme has to be degraded from our body and removed. Now heme is degraded into a molecule called biliverdin, which gets converted into bilirubin. This bilirubin is insoluble, so it forms a complex with albumin in order to be transported to the liver, and then it is passed through the bile ducts and the intestines, where it is converted to stercobilinogen and urobilinogen, which are colorless compounds, but they are oxidized. So urobilinogen is transported to kidney, and it is oxidized in kidney, and stercobilinogen is oxidized in the uh, large intestine, and it is converted into stercobilin. Stercobilin and urobilin both are excreted in the feces and urine, respectively. So this is how the conversion occurs. So bilirubin, I told you, in the liver gets converted into bilirubin diglucuronide by the enzyme glucuronal bilirubin transferase. So this bilirubin diglucuronide again is transported to the intestine, and in the bile it is secreted, and it gets converted into urobilinogen. Urobilinogen is transported to kidney, where it is converted to urobilin. and urobilinogen in the bile and in the intestine only, it gets converted to stercobilinogen and stercobilin. Okay, so this is urobilin, this is stercobilin. I hope that's clear. Moving to the next question now. Hyperbilirubinemia occurs when bilirubin exceeds. Hyperbilirubinemia occurs when bilirubin exceeds option A, 0.5 mg per dl, option B, 1 mg per dl, option C, 1.5 mg per dl, and option D, 2.5 mg per dl. Hyperbilirubinemia occurs when bilirubin exceeds. Two point 0.5 mg per dl, 1 mg per dl, 1.5 mg per dl, or 2.5 mg per dl. So the correct answer is option B, 1 mg per dl. So hyperbilirubinemia, by the name itself, you can see, you can understand bilirubin is excess in, and it is now present in the plasma. So plasma bilirubin levels increases. So it leads to hyperbilirubinemia. Okay. So normal plasma bilirubin levels ranges from zero point 0.2 to 0.8 mg per dl. The out of which the unconjugated bilirubin is 0.2 to 0.6 mg per dl and conjugated bilirubin is 0 to 0.2 mg per dl. Okay. This if the plasma bilirubin level exceeds 1 mg per dl, the condition is called hyperbilirubinemia. And the levels between 1 and 2 mg per dl are indicative of latent jaundice. And when the bilirubin level exceeds 2 mg per dl, it diffuses into the tissues, producing yellowish discoloration of sclera, uh, conjunctiva, skin, and mucous membrane, resulting in jaundice. So, if you have ever suffered from jaundice, or if you have seen anyone suffering from jaundice, you would have seen their eyes turning yellow, their skin turning yellow. That is due to the excess bilirubin levels and that diffuses into the tissues. So when the plasma bilirubin level increases, it diffuses into the tissues, leading to jaundice. Okay. I hope that's clear. The next question. Krigler-Najjar syndrome is a rare genetic disorder that results in complete absence of bilirubin conjugation. Option B, impaired bilirubin uptake by the liver. Option C, impaired bilirubin excretion into the bile. Option D, increased bilirubin production. Krigler-Najjar syndrome is a rare genetic disorder that results in option A, complete absence of bilirubin conjugation. Option B, impaired bilirubin uptake by the liver. Option C, impaired bilirubin excretion into the bile. Option D, increased bilirubin production. The correct answer is option A, complete absence of bilirubin conjugation. So Krigler-Najjar syndrome, it's a rare genetic disorder, and it is due to the complete absence of bilirubin conjugation. So if you remember from the bilirubin excretion uh, process, the bilirubin is conjugated with albumin, right? Because bilirubin is not soluble in water, it is insoluble. And for the transport of bilirubin through the plasma to the liver, it needs to be solubilized somehow. So it is conjugated with albumin, and albumin carries the bilirubin from the plasma to the liver, where it is further processed and excreted through urine and feces. Okay. So if this syndrome, Krigler-Najjar syndrome, this is a genetic disorder which results in absence which results due to the absence of this bilirubin conjugation. So as a result, bilirubin levels in the plasma increases because there's no conjugation with albumin, thus there's no transport to the liver. Okay, and it can lead to jaundice-like symptoms. But this is a genetic disorder. This is not an acquired disease. Okay, I hope that's clear. Moving to the next question. Dubin-Johnson syndrome is characterized by Dubin-Johnson syndrome is characterized by option A, reduced conjugation, option B, impaired excretion of conjugated bilirubin, option C, increased hemolysis, and option D, impaired bile acid synthesis. Dubin-Johnson syndrome, it is characterized by option A, reduced conjugation, option B, impaired excretion of conjugated bilirubin, option C, increased hemolysis, and option D, impaired bile acid synthesis. What do you think would be the correct answer? The correct answer is option B, impaired excretion of conjugated bilirubin. So Dubin-Johnson syndrome, it is characterized by the impaired excretion of the conjugated bilirubin. So in the previous question, we had Crigler-Najjar syndrome, which was due to the absence of the conjugation, bilirubin conjugation. Okay. In this case, in Dubin-Johnson syndrome, the conjugation is happening. Bilirubin and albumin conjugation is happening, but it is not being excreted via liver and bile. Okay, so there's a defect in the excretion part of the conjugated bilirubin, and as a result, conjugated bilirubin increases in the system. I hope that's clear. Moving to the next question now. In which type of jaundice is bilirubin absent from urine? In which type of jaundice bilirubin is absent from urine? Option A, hemolytic jaundice. Option B, hepatic jaundice. Option C, obstructive jaundice. And option D, Rotor syndrome. In which type of jaundice is bilirubin absent from urine? Hemolytic jaundice, hepatic jaundice, obstructive jaundice, and option D, Rotor syndrome. Which type of jaundice is bilirubin absent from urine? Hemolytic jaundice, hepatic jaundice, obstructive jaundice, or Rotor syndrome? The correct answer is hemolytic jaundice. That is option A. So hemolytic jaundice, it's a hemolytic disease of the newborn, and it actually is caused due to the erythroblastosis fetalis. So what happens is, so we have two types of blood type, Rh positive and Rh negative, right? So you all know about the blood groups A, B, and O, and we have A positive, A negative, B positive, B negative, O positive, O negative, and AB positive, AB negative, right? So these are due to the presence and absence of certain antigens. Okay. Now, this positive and negative is due to the presence and absence of the Rh antigen. Okay. The name comes from the rhesus monkey. So, Rh antigen, presence and absence of the Rh antigen denotes the blood type, either it is positive or negative. Now, let's say the mother is Rh negative. Mother has a blood type which is negative blood type, and the father has a positive blood type. Okay, so when they conceive, sometimes there's a chance that the fetus might have a positive blood type. Okay, now if the mother has a negative blood type, mother does not have the Rh antigens, that's why it is Rh negative. Now the fetus, if it is Rh positive, the Rh antigens present in the fetus would be foreign to the mother's body, due to which what will happen, the mother will start developing antibodies against the Rh antigens. And during the first time pregnancy, this problem, like, it doesn't cause any major problem, this Rh incompatibility, because at that stage the mother is producing the antibodies against the Rh antigen, right? But in the second pregnancy, what happens now, the mother has the Rh antibodies because the body has been exposed to Rh positive antigen. So body has now prepared antibodies, and now if in the during the second pregnancy again the fetus is Rh positive, now the mother's Rh antibodies will start attacking the fetal red blood cells, okay, because they have the Rh antigen. And now for the mother's body, those antigens are foreign particles against which antibodies have been raised, so it's a defense mechanism, right? Antibody production in our body is a defense mechanism. So mother's antibodies are going to fight the fetus red cells. Okay. Now, what will happen? It will start destroying the fetal red cells even before birth. And this could lead to severe hemolytic diseases in the child, often lead to erythroblastosis fetalis. Okay. And one such hemolytic disease is hemolytic jaundice, where uh, the blood level is more than 20 mg per dl, and the capacity of albumin to bind bilirubin is exceeded. And in young children before the age of 1 year, the blood brain barrier is not fully matured. So, if more and more bilirubin is uh, getting accumulated, it can actually cross this blood brain barrier because it is not fully matured in those young kids, and that could lead to free bilirubin entering the blood, which the condition is called kernicterus. And it is since when bilirubin starts depositing in blood, it can lead to mental retardation, fits, toxic encephalitis, and spasticity. Okay, so it can cause all kinds of uh, mental retardation symptoms, and uh, it can it can be very toxic to the brain. Okay. And if the child develops hemolytic diseases, child may be given exchange transfusion along with phototherapy and barbiturates. Okay. So phototherapy with blue light isomerizes the insoluble bilirubin. So now I told you the bilirubin was insoluble. That's why that conjugation with albumin was necessary. And now excess of bilirubin has been produced. So it has to be solubilized somehow. So exposure of the young child with blue light at 440 nanometer actually isomerizes the insoluble bilirubin to make it more soluble. And once it is soluble, it can be excreted out through urine, right? So even without conjugation, it can be excreted out. So conjugation was occurring for making it solubilized. Now, if the conjugation is not possible due to high concentration of bilirubin presence, in that case, we need to use other methods to make bilirubin solubilized. So phototherapy is one such method where the child is exposed to the blue light at 440 nanometer wavelength, which isomerizes the insoluble bilirubin to more soluble isomers. Okay, I hope that's clear. Moving to the next question. Which pigment gives urine its yellow color as a result of bilirubin metabolism? Which pigment gives urine its yellow color as a result of bilirubin metabolism? Option A, stercobilin. Option B, urobilin. Option C, biliverdin. And option D, hemine. We have already discussed this question. Which pigment gives urine its yellow color as a result of bilirubin metabolism? Option A, stercobilin. Option B, urobilin. Option C, biliverdin. And option D, hemine. So the correct answer we have already discussed. This is option B, urobilin. So biliverdin, that comes from heme degradation. It is green in color. It turns into bilirubin, which is yellow in color, and bilirubin is excreted out in the form of urobilin and stercobilin. Urobilin is yellow in color and gives the urine yellowish color, and stercobilin is red brown in color and it gives the color to the feces. Okay. So during jaundice, I've already explained the yellow skin and eyeball is due to the impaired liver function, which leads to the bilirubin accumulation and eventual bilirubin presence in the tissues. Moving to the next question now. In obstructive jaundice or post-hepatic jaundice, which of the following is typically observed? In obstructive jaundice or post-hepatic jaundice, which of the following is typically observed? Option A, increased unconjugated bilirubin only. Option B, pale stools and dark urine. Option C, increased urobilinogen in urine. And option D, hemolysis. In obstructive jaundice or post-hepatic jaundice, which of the following is typically observed? Increased unconjugated bilirubin only. Option B is pale stools and dark urine. Option C, increased urobilinogen in urine. And option D, hemolysis. So the correct answer is option B, pale stools and dark urine. So in case of obstructive jaundice, what happens? The conjugated bilirubin is increased in the blood and it is excreted in the urine. So if there is complete obstruction, UBG will be decreased in the urine and even absent. So you know the color of the urine was due to the presence of UBG or urobilin. Uh, so if urobilin is absent, then the uh, the color of the urine will be colorless or transparent, right? And in total obstruction of biliary tree, the bile does not enter the intestine. So this pathway will also not happen and there will be no pigment. Then there will be no stercobilin, which will result in the feces uh, to be clay colored. So pale stools and dark urine is the correct answer. So pale stool due to sorry, urine won't be colorless. Urine will be dark in color. Pale stools due to the presence of due to the absence of stercobilin, and dark urine because the conjugated bilirubin now will be directly excreted out, which will result in the dark colored urine. Okay, so there won't be any UBG present. There won't be any conversion of conjugated bilirubin into urobilin. So that results in the that happens in the obstructive jaundice. Okay. Moving to the next question. PRPP is important in nucleotide biosynthesis because option A, it is produced from uric acid. Option B, it is a precursor of both purines and pyrimidines. Option C, it inhibits IMP dehydrogenase. And option D, it is the end product of purine catabolism. PRPP is important in nucleotide biosynthesis because it is produced from uric acid. Option B, it is a precursor of both purines and pyrimidines. Option C, it inhibits IMP dehydrogenase. And option D, it is the end product of purine catabolism. What could be the correct answer here? The correct answer is option B. PRPP is important in nucleotide biosynthesis because it is a precursor for both purines and pyrimidines. Okay, so it is the first step in the nucleotide biosynthesis, which is common in case of both purine and pyrimidine synthesis. And that's why it is called the preparatory step, or it is numbered as step 0 because it is common in both purine and pyrimidine synthesis. So when we study the purine synthesis, we'll start from the step 1, which occurs after PRPP. And for pyrimidine synthesis also, the step 1 would be after PRPP. So step 0 is a common step between both purine and pyrimidine synthesis. So this first step in the nucleotide biosynthesis is basically PRPP synthesis. What is PRPP? PRPP is phosphoribosyl pyrophosphate. Okay, phosphoribosyl pyrophosphate. So now from the name itself, you can see what all you require as a substrate. Phosphoribosyl, so you need a ribose sugar attached to a phosphate group. So ribose 5-phosphate, and pyrophosphate will come from the ATP. Okay, so we need ribose 5-phosphate and ATP as the starting material. So ribose 5-phosphate and ATP can together form phosphoribosyl pyrophosphate and AMP, because pyrophosphate will be removed from ATP. So ATP will be converted to AMP. The enzyme that is responsible for this reaction is ribose phosphate pyrophosphokinase. So, phosphate transfer reactions are catalyzed by the enzymes kinase, right? So, ribose phosphate, because ribose phosphate is there, and what is being transferred? Pyrophosphate is transferred. So, pyrophosphokinase, okay? And it will produce phosphoribosyl pyrophosphate, okay? So, ribose 5-phosphate and ATP together with the enzyme ribose phosphate pyrophosphokinase, or it is also known as PRPP synthetase. And why synthetase? Because ATP hydrolysis is uh, required. So ATP is basically the phosphate group donor here. So ribose group already has a phosphate attached to the 5th carbon. Ribose is a 5-carbon sugar, right? So 1, 2, 3, 4, and 5th carbon is here. And the phosphate group is attached to the 5th carbon. So it is ribose 5-phosphate. And then by the enzyme PRPP synthetase, one ATP molecule gives 2 phosphate groups, a pyrophosphate group, uh, to this ribose 5-phosphate. And the pyrophosphate gets attached to the 1st carbon. Okay, so it is phosphoribosyl, because the this phosphoribosyl, the phospho in the phosphoribosyl denotes to the original phosphate that was present in the ribose at the 5th carbon. Pyrophosphate is attached to the 1st carbon. Okay, so this you have to remember. It's not a ribose triphosphate or something. It is the phosphate groups are attached to different carbon. The first phosphate group is attached to the 5th carbon, so ribose 5-phosphate. And when the pyrophosphate from ATP is transferred, uh, it becomes phosphoribosyl pyrophosphate. The enzyme involved is PRPP synthetase. Synthetase because ATP hydrolysis is involved. How ATP hydrolysis is involved? ATP is the one that is giving the pyrophosphate to the ribose 5-phosphate, and in turn getting converted to AMP. Okay. So ribose 5-phosphate plus ATP is becoming phosphoribosyl pyrophosphate plus AMP by the enzyme ribose phosphate pyrophosphokinase or PRPP synthetase. And this PRPP is also used for synthesis of pyrimidine nucleotide. I have mentioned this is used for both purine and pyrimidine synthesis and for the salvage pathway. So there are 2 pathways, de novo pathway and salvage pathway. We'll discuss that eventually and in the next session as well. So PRPP is not considered as a step in the de novo synthesis of purine nucleotide and is called preliminary step or preparatory step or step 0. I already explained you why. Let's see the next question. What is the committed or rate limiting step in the de novo purine biosynthesis? What is the committed or the rate limiting step in the de novo purine biosynthesis? Option A, conversion of IMP to AMP. Option B, synthesis of PRPP. Option C, amido transfer from glutamine to PRPP. And option D, synthesis of GMP. What is the committed or rate limiting step in the de novo purine biosynthesis? Option A, conversion of IMP to AMP. Option B, synthesis of PRPP. Option C, amido transfer from glutamine to PRPP. And option D, synthesis of GMP. The correct answer is option C, amido transfer from glutamine to PRPP. So this is the first step in the de novo uh, biosynthetic pathway of purine. So you saw in the step 0 or the preparatory phase, we got the PRPP, phosphoribosyl pyrophosphate. This phosphoribosyl pyrophosphate reacts with glutamine, where enzyme called phosphoribosyl amidotransferase acts on it, and the amine group of glutamine is transferred to the PRPP, and the pyrophosphate is released. Okay, so there's there's a breakdown of the pyrophosphate bond. Pyrophosphate is released. The amine group is transferred to this carbon where the pyrophosphate is attached to this carbon itself. The amine group is now attached, transferred from glutamine. And once amine group is transferred from glutamine, what will glutamine become? Glutamine will become glutamate or glutamic acid, right? So the amine group from the glutamine will be transferred to PRPP by the enzyme phosphoribosyl amidotransferase. So amidotransferase, there's a transfer of amine group to PRPP, resulting in the formation of PRA or 5-phosphoribosyl 1-amine. So already at the 5th position, the phosphate was always there because we started from ribose 5-phosphate, right? So now the product that we are getting is 5-phosphoribosyl at the first position where pyrophosphate was attached in PRPP. Now in that first position, amine is attached. So we get 5-phosphoribosyl 1-amine because amine is attached to the first carbon. So 5-phosphoribosyl 1-amine. And it is also known as PRA. It is abbreviated as PRA. So from PRPP, phosphoribosyl pyrophosphate, we get the product PRA, and this is the first step in the purine biosynthesis pathway. The enzyme that catalyzes this reaction is PRPP amidotransferase, and the amine group donor here is glutamine. And glutamine, once it donates the amine group to PRPP, it becomes glutamate or glutamic acid. Okay. I hope that's clear. Moving to the next question, the regulation of the de novo purine synthesis pathway is primarily controlled by the feedback inhibition of which end product? The regulation of de novo purine synthesis pathway is primarily controlled by the feedback inhibition of which end product? And multiple answers could be correct in this question, so be mindful of that. Option A, adenosine monophosphate or AMP. Option B, guanosine monophosphate or GMP. Option C, inosine monophosphate or IMP. And option D, hypoxanthine. The regulation of de novo purine synthesis pathway is primarily controlled by the feedback inhibition of which end product? AMP, GMP, IMP, or hypoxanthine? Regulation of de novo purine synthesis pathway is primarily controlled by the feedback inhibition of which end products? The correct answer is option A and option B, that is AMP and GMP. So AMP and GMP can both uh, regulate the de novo purine synthesis by feedback inhibition. So these are the end products. So if you remember the feedback inhibition from heme synthesis, the heme was the end product and it was inhibiting the starting enzyme. and controlling the synthetic pathway. Similarly, in the purine synthesis, AMP and GMP are the end products, one of the end products, and uh, these AMP and GMP end products, they inhibit the uh, enzymes involved in the de novo purine synthesis pathway and regulate the biosynthetic pathway. So the committed step in de novo synthesis is the reaction catalyzed by amidotransferase 1. I told you the first reaction, that is from PRPP, we get PRA, phosphoribosylamine. So this is the rate limiting step or the committed step, right? So if you inhibit this enzyme, uh, the pathway can be easily regulated. Okay. So this enzyme is inhibited by AMP and GMP, which are the end products. So formation of AMP from IMP requires GTP. So you see here, we had ribose 5-phosphate forming PRPP, right? And PRPP gets converted to PRA by the enzyme PRPP amidotransferase, right? And this is the rate limiting step. Now that PRA gets converted into IMP, IMP gets converted into AMP and GMP, and AMP eventually gets converted into ATP, GMP gets converted into GTP. Now, the important thing to note that ATP is required for the GMP formation and GTP is required for the AMP formation. Okay, so this formation of formation of AMP and GMP from IMP. This is required by the end products of these individual pathways. The GTP is required for AMP synthesis and ATP is required for GMP synthesis. Okay, so this is one type of control. And both this AMP and GMP, they can inhibit the PRPP amidotransferase that was catalyzing the rate limiting step. So they both, AMP and GMP, they both can inhibit this enzyme, and this would be a type of feedback inhibition. Okay, so both AMP and GMP inhibit their own formation by feedback inhibition of their formation from IMP, and also they also inhibit FGPRT. Okay, so this is how the purine synthesis is regulated. This is the de novo purine synthesis pathway. I hope that's clear. This is the last question of today's session. So I hope all the topics have been covered and through the multiple questions in this session. So these questions will help you solve the assignment questions for this week. So with this, I'll end the session now. Thank you so much for listening to the session.